Hello friends, good morning, good afternoon and good evening. I am Sitaram Wahule and I have been teaching biology right from the last 30 years. Now I wish you all warm welcome at Web Education, that is learning through understanding of textbook of State Board of Maharashtra, textbook of CBSE and textbook of ISC as well. Well, in the last lecture we have studied the human health and introduction of immunity system from the topic of human health and diseases. Today we are going to study the part next to that, that is the first line of defense, physical and chemical barriers of innate immunity system from the same topic, that is the human health and diseases. So my dear friends, without wasting time, let's get started.
Textbook of Maharashtra, innate immunity or inborn immunity. See, it has been described here in this paragraph. Innate immunity is the resistance to infection that an individual possesses by virtue of his or her genetic makeup. It means that the innate immunity system is hereditary. That is, it can be transmitted from one generation to another generation. For example, if a parent has certain genetic deficiencies in this system, then that can be passed to the child as an inheritable immune deficiencies. Then next to this, it is the natural, that is inborn defense system of the body. It is not affected by prior contact with microorganisms or immunization. That is, the innate immunity system retains no memory. Then next to this, it has been mentioned over here, it is non-specific when it indicates a degree of resistance to infection in general, or specific where resistance to a particular pathogen is concerned. Now in order to understand this concept, so my dear friends, I would like to repeat it once again, that is, the innate immunity is non-specific when it indicates a degree of resistance to infection in general, or a specific where resistance to particular pathogen is concerned. It means that the author is trying to explain the dual nature of the innate immunity system, that it is non-specific as well as specific. Now how can we say that the innate immunity system is specific? We study this small concept. So here you will find recognition of pathogens, that is microbes, in innate immunity system. I have written over here PAMPS, the long form of this, pathogen associated molecular patterns. At the same time I have written over here DAMPS, that is damage associated molecular patterns. Now this damage associated molecular patterns, they are found or they are released by the host cell which are damaged, or the cell which are injured, or the cell they are having the tumors. So these cells, whoever having the tumors or the cells which are damaged or injured, these are the host cells which releases certain signaling molecules, and that signaling molecules are called as damage associated molecular patterns. Then the concepts here we have to learn is PAMS, that is pathogen associated molecular patterns. So these pathogen associated molecular patterns is located on antigens, that is the foreign bodies. Now in order to identify this PAMS, there are certain PRRS, that is pattern recognition receptors present on the surface of the immune cells or inside of the immune cells or in the body fluids as well. So these are the PRRS, these are present on the immune cells. And PAMPS, these are present on the foreign bodies, that is the antigens. So what are the examples of the PAMs over here? In gram negative bacteria, lipopolysaccharides, that is LPS is present. And number 2, in gram positive bacteria, lipoic acid and peptidoglycan is present. Then number 3, in flagella of bacteria, the protein flagellin is present. And in viruses, the genetic material is present in the form of double-stranded RNA or single-stranded RNA. So these are called as PAMs, is pathogen associated molecular patterns. Now these pathogen associated molecular patterns, they are identified by means of pattern recognition receptors. They are present on the surface of the immune cell, as I already told you all, or some of them they are present intracellularly, and some of them they are also present in the body fluids as well. Now what are the types of the PRRS? Number 1, the TLRS, that is toll-like receptors. So there are 10 toll-like receptors are present in the case of the human being. Number 2, CLRS, that is C-type lectin receptors. About 15 CLRS are present in the human. Number 3, RLRS, that is Reg-like receptors. Number 4, NLRS, that is node-like receptors. And number 5, secretory or complementary receptors. So these are present in the body fluids. So out of this, the first 4 are very important. Out of this first 4, the first 2, that is TLRS and the CLRS, they are called as the surface receptors. And the third and fourth, generally they are called as the intracellular receptors, since they are present within the cells, inside the cells of the components of the immunity system. So out of these 5, so I am going to explain to you all the first one, that is toll-like receptors. So my dear friends, how is the name toll came into being? So toll is nothing but it is a particular type of gene which is found in Drosophila melanogaster, a fruit fly. So this toll gene in the fruit fly not only controls the embryonic development of the fruit fly but also protects it from the infection. The same types of the receptors, they are there in the components of the immunity system. So as I have already told you all, so in the case of the human, there are 10 toll-like receptors are present. Now see the pathogen associated molecular patterns, they are not present in the eukaryotic cells, including human. They are found only in the antigens. So the toll receptors number 4 is associated with gram-negative bacteria, that it identifies the lipopolysaccharide which is present in the cell wall. Then toll-like receptors number 2 is associated with gram-positive bacteria. It means that identifies the peptidoglycan which is present in the cell wall of the gram-positive bacteria. Then Toll-like receptors number 5, which identifies the flagellin in the flagella of bacteria, and toll receptors number 3, which identifies or which recognizes the double stranded RNA, and toll-like receptors number 7 and 8, which recognizes the single stranded RNA. Now, as I have already told you all, the toll-like receptors number 4, which identify the lipopolysaccharides only, and this lipopolysaccharide is shared by number of microorganisms. So what I meant to tell to you all here is that the components of the innate immunity systems, they are not able to recombine and mutate so as to recognize the spectacular range of molecular patterns which can be easily recognized by the B cells and the T cells of the adaptive immunity. So my dear friends, I would like to say that because of all these factors, we can say that the innate immunity system is less specific when compared to the adaptive immunity system. Next to this, we are going to study that here it has been written, one form of innate immunity comprises the various types of barriers which prevent the entry of foreign agents into the body. Epithelial surface. So my dear friends, under the heading as a epithelial surface, so whatever the organs, then the tissues and the chemicals we are going to study over here, they are collectively called as the components of first line of defense of the innate immunity system. Well, all these components of the innate immunity system have to encounter the pathogen in the atmosphere on one hand and to maintain the healthy body on the other hand. Then these organs, the tissues and the chemicals of the innate immunity system acts as barriers, thereby fighting and not allowing any pathogen to enter inside the cell or in the bloodstream as well. So my dear friends, under the heading as the epithelial surface, here you will find I have highlighted some of the contents with the gray color and some of the contents with the light pink color. The contents which is highlighted by me with the gray color constitute the physical barriers, and the contents which is highlighted by me here with pink color constitute the physiological barriers. Now let us study one by one. The first one, the contents which is highlighted by me with gray color. Here it has been mentioned as the intact skin and the mucus covering the body protect it considerably against invasion by microorganisms. So as far as the physical barriers of the innate immunity system is concerned, only two examples have been given over here. Number one is the skin and the number two is the mucus covering the body. So dear friends, skin is the organ and the mucus covering is the tissues. Now let us study one by one. The first one is the skin. The skin is the largest organ of all the organ in the human body, and it is made up by means of two layers. That is, the upper layer is called as the epidermis and the lower layer is called as the dermis. Let's we study the basic structure of the skin. See my dear friends, this is the diagram showing the structure of the human skin. As I have already told you all, the human skin is made up by means of two layers. The uppermost layer is called as the epidermis. This one is the epidermis, and I have highlighted, I have magnified the part of the epidermis over here. Then below the epidermis there is a dermis. So this part of the skin is called as the dermis. This part is called as the dermis, and the dermis is made up by means of connective tissue, then the blood vessels and the nerve fibers. Now see the epidermis. This region is called as the epidermis, and I have magnified over here. As the epidermis of the skin of the man or human is made up by means of many layers. You will find right from bottom to the surface. So the layer which is present at the bottom is called as the basal cell layer. This layer is also called as stratum germinativum. Then the layer which is present here is called as the stratum spinosum, this layer. Then which is followed by the stratum granulosum. This layer of the cell is called as stratum granulosum, and the surface layer of the cell is called as the stratum corneum. So it means that there are 4 layers. The epidermis is made up by means of 4 layers starting from bottom to the surface. As I have already told you all that the basal cell layer is also called as the stratum germinativum or Malpighian layer. It rests on the basement membrane, and this layer has got the property of multiplication. The cells here, they are columnar in shape and divides and redivides continuously so as to produce new and newer cells. As the new cells, they are produced by the basal cell layer or the stratum germinativum. They are continuously pushed to the surface, and when they migrate from bottom to the surface, certain changes takes place, not only in their shape but their composition also. So as far as the shape of the cell is concerned, when the cells are newly formed, they are columnar in shape. When they reach in the middle, they become circular and oval, and by the time they reach to the surface, they become flat. And the cytoplasm is completely replaced by means of the dead protein, and that dead protein is called as the keratin. It means that, and because of that keratin, the skin of man is very hard. Since the skin is very hard, thereby it does not allow any microorganism to penetrate unless and until it is broken. But in case if any microorganism adheres over here to the uppermost layer of the skin and trying to pose the potential harm to the body of the animals, at that time those microorganisms which adhered over here, they are dislodged or removed, as the uppermost layer of the cells, they are periodically shut up or filled up as the cells grow old and die. So my dear students, here you will find, as far as the skin of the animal is concerned or the human is concerned, that the uppermost layer of the human skin is called as stratum corneum, and it contains the keratinized cells. It means that they are filled by means of the dead protein, and that dead protein is called as the keratin, and because of that the uppermost layer of the skin becomes very hard, and because of that the skin does not allow any microorganism to penetrate inside the body. Similarly, as one of the important property of the epithelial tissue is that its uppermost layer of the cell is continuously set up or filled up as the cells grow old and die, and because of that it does not allow any microorganism to settle on the surface of the skin. Now see the second example of the physical barrier. It has been explained over here as the mucus covering the body. So what are the areas of the body where you will find the mucus covering? For example, all the orifices which communicates to the exterior contains they are lined by means of mucus covering. For example, the oral cavity, gastrointestinal tract, then respiratory tract, then urogenital tract, then the eyes as well as ears. There you will find the mucus covering the body. Now this mucus covering of the body is also lined by means of the epithelial surface, as we have studied in the case of the human skin, that epidermis is made up of by means of many layers of the cells. They are arranged one above the other. That is that the epidermis of the human skin is nothing but is a compound epithelium. The same condition applies over here also in the case of the mucus covering the body. So it is also lined by means of compound epithelium, and somewhere it is lined by means of the simple epitheliums. So in the case of the gastrointestinal tract, particularly along the lining of the stomach and intestine, that the mucus covering, they are lined by means of simple epithelium, that is a single layer of columnar epithelium. Excluding these two parts, that is the stomach and intestine, then the most parts of the respiratory tract, then the most parts of the urogenital tracts, as well as in the eyes and in the ears, that the mucous coverings made up by means of epithelium, and that is the examples of the compound epithelium, and their structure is more or less similar to that of stratified squamous epithelium, as we have studied in the case of the human skin. So my dear students, the main function which is performed by the mucus covering the body is nothing but to trap the microorganism, and this is how they are prevented from entering inside the main organs of the body. Now here you will find the mucosa of the respiratory tract has been explained here. See, the mucosa of the respiratory tract has several innate mechanisms of defense. The nose prevents entry of microorganisms to large extent. The inhaled particles being arrested through the hair at or near the nasal orifices. So friends, you all are knowing that the inside the nose the nasal cavities are present, and the external openings of the nasal cavities are called as the external nares, and the internal openings of the nasal cavities are called as the internal nares, and the portion which is present between the external nares and the internal nares, that constitute the nasal cavities. And the nasal cavities, they are divided into three parts. The anterior one is called as the vestibule, the middle one is called as the respiratory part, and the innermost is called as the sensory parts. And the sensory part is lined by means of sensory epithelium. That's why it plays very important role in detection of the smell. The middle part is called as the respiratory part, and since it is lined by means of many blood capillaries, that's why it plays very important role to warm the air before going into the lungs. And the anterior most parts of the nasal cavities, which is present over the external nares or the nostrils, is called as the vestibule. So that vestibule part of the nasal cavities is lined by means of mucus coated hair, as well as the epithelium, which is also coated with the mucus. Then these two parts, the mucus coated epithelium as well as the mucus coated hair, traps the dust as well as microbes which happens to enter inside the lungs along with the inhaled air. But there are chances that some of the microbes along with the dust may find their entry into the trachea or the bronchi, where they are subjected to be entrapped or entangled by the mucous lining of the trachea. Once they are entrapped or entangled by the mucous lining of the trachea, and thereby what happens, the outward rhythmic ciliary movements push them back into the pharynx, and from the pharynx those microorganisms, they are thrown outside the body through the process of coughing or sneezing, or maybe swallowed and digested into the stomach. Now comes toward the physiological barriers of the innate immunity system. The contents which is highlighted over here with light pink color shows the physiological barriers. Now what are the examples we are going to study under the physiological barriers are as follows. Number 1, sweat. Number 2, sebum. Number 3, mucus. Number 4, saliva. Number 5, hydrochloric acid, HCl. Next, the urine. Then the next, semen. Then the next, tears. The example is given over here. The skin possesses bactericidal activity due to the presence of a high concentration of the salt in the drying sweat, and the sebaceous secretions and long chain of fatty acids have bactericidal and fungicidal properties. So in this para, the role of sweat and the sebum has been explained over here. As you are knowing that the sweat is the product of the sweat glands. They are also called as the sudoryphilic glands, and the sebum is the product of the sebaceous glands. So both of these glands, they are found in the dermis, but they open to the surface of the skin, that is on the epidermis. So what is the composition of the sweat? The sweat is composed of by means of water, then NaCl, then lactic acid, certain minerals are also there, and urea in traces. Then what is the composition of the sebum? That the sebum is composed of by means of glycerol, then the fatty acids, then the wax, and the cholesterol. Now the fatty acids from the sebum and the lactic acid from the sweat plays very important role to maintain the low pH on the surface of the skin, and thereby it avoids the multiplication of the microorganisms on the surface of the skin. Then the sebum is the product of the sebaceous glands, and the sweat is the products of the sweat glands. Both of them, they mix to each other so as to form very thin film on the surface of the skin, and that thin film of the sweat mixed with the sebum, it has got bactericidal and fungicidal properties. Bactericidal means to kill the bacteria, and fungicidal means to kill the fungi. As you are knowing that the sweat glands, they are scattered throughout the entire skin. They are the simple tubular glands. They are located in the dermis, but they open to the surface by means of a small pore. That small pore is called as the sweat pore. Though the sweat glands, they are scattered throughout the entire skin, but their percentage increase on certain parts of our body. For example, it is the forehead, then tip of the nose, then armpit, then palm of the hands, and the soles of the feet, where you will find the maximum number of the sweat glands. And the sebaceous glands, they are also located in the dermis. They are the compound glands. The same types of the glands you will find in the ear also, and secrete the same product that is called as sebum. But the type of the sebaceous glands, they are located in the ear, they are called as the ceruminous glands, and the type of the product which is secreted by the ceruminous glands is called as the cerumen, having the same composition. So dear student, sweat also contains an antimicrobial enzyme that is called as the lysozyme. That lysozyme is active against the gram-positive bacteria. It brings about the degradation of the gram-positive bacteria. Next to this, it has been mentioned over here, as per the physiological barriers of the innate immunity system is concerned, the mouth is constantly bathed in the saliva, which has inhibitory effect on the microorganisms. This sentence has been given regarding the saliva, and you are knowing that saliva is secreted by means of the 3 pairs of the salivary glands, 1 pair of the parotid glands, 1 pair of submandibular glands, and 1 pair of sublingual glands. The major source of the saliva in the oral cavity is the salivary glands. The saliva also contains the lysozyme, which is active against the gram-positive bacteria only. Then, so my dear friends, you should know that our oral cavity harbors more than 700 different species of the bacteria. The most common species of some of the microorganisms which are there always in our oral cavity are as follows. The bacterial species, number 1, the Streptococcus mutans, number 2 Actinomyces species, and number 3 Porphyromonas gingivilis. The species of the fungi which is common in our oral cavity is Candida albicans, and the species of the protozoan which is common in our oral cavity is Entamoeba gingivilis, and some of the species of the viruses they also found in our oral cavity is herpes virus and cytomegalovirus. Next to this, it has been mentioned over here, the acidity of the gastric secretion in the stomach destroys microorganisms. So in the stomach, as you are knowing that hydrochloric acid is present, which is secreted by the parietal cells of the gastric glands. Then the chief cells secrete the pepsinogen and the mucous cells secrete the mucus. Then the D cells of the gastric gland secretes somatostatin and the G cells of the gastric gland secrete gastrin. We have already studied during the 11th standard. Now this acidity of the gastric secretion in the stomach destroys all the microorganisms. Then the flushing action of urine eliminates bacteria from the urethra, and spermine and zinc present in the semen are antibacterial. The conjunctiva is freed of foreign particles by flushing action of lacrimal secretions. So here we have to study the role of tears, which is secreted by the lacrimal glands. The tear also contains the lysozyme enzyme, which is active against the gram-positive bacteria. The lysozyme, for the first time described by the Alexander Fleming. The lysozyme is thermolabial. It means that its action depends on a certain ranges of the temperature. Then it is low molecular weight. It is a basic protein which acts as a muramidase. So muramidase is nothing but it is a lysozyme. So my dear students, this is how we have studied the physical and physiological barriers of the innate immunity system from the textbook of Maharashtra. So now let's proceed toward the textbook of CBSE. CBSE textbook. See the physical barriers and the physiological barriers. Today we are dealing with only two barriers only. Now as per the physical barriers, it has been mentioned here, skin on our body is the main barrier which prevents entry of the microorganism. Mucous coating of the epithelium lining the respiratory, gastrointestinal and urogenital tracts also help in trapping the microbes entering our body. So my dear students, we have already studied every details regarding the skin as well as the mucous coating of the epithelial lining of the respiratory, then the gastrointestinal and the urogenital tracts. We have already studied every details as per the physical barriers which are explained in the textbook of State Board of Maharashtra. Next to this, the physiological barriers. So as per the physiological barriers in the textbook of CBSE has been mentioned, the acid in the stomach, saliva in the mouth. The saliva in the mouth also contains the lysozyme enzyme. Then tears from the eyes all prevent microbial growth. So we have already studied every details. Textbook of ISC, latest publication by Nutan. First line of defense of innate immunity system. We are studying physical barriers and the physiological barriers. See the physical barriers and the physiological barriers, they are explained in the textbook of ISC are as follows. Now the first one is the physical barriers. Skin on our body is the main barrier which prevents entry of the microorganism. Mucous coating of the epithelium lining the respiratory, gastrointestinal and urogenital tracts also helps in the trapping microbes entering our body. We have already studied every details. It was already told to you all. Then the physiological barriers, acid in the stomach, saliva in the mouth and tears from the eyes all prevent microbial growth. So physiological barriers also we have already studied. Now it is here that I would like to stop my today's video lecture. Now, so far you did not hit the like button, so I request you all to hit the like button and subscribe my channel so that I would prepare the next video lecture on the second line of defense of the innate immunity system. Till that time, bye bye to you all.